Hi, good morning. This is Kavita Kundra, and today we are covering IGNOU MSCDFSM Applied Physiology, Unit 2, Cell and Blood. We all know about cell, and we are starting cell from the very beginning. It is a small self-functioning unit found in all living organisms, and each cell is enclosed by an outer membrane or a wall called cell membrane. A cell has a receptor on its surface. It contains genetic material that is called DNA. Nucleus is present inside the cell and cytoplasm. Cell together form tissue. Tissue forms organ, then organ system, and then it together form organism. What is totipotency? We have heard this word a lot, but what exactly it is? It is the potential of cell to give rise to unlike cell is called totipotency. Discovery of the cell, Robert Hooke, 1665, he observed honeycomb-like structure consisting of thin wall and compartment called cellulae, renamed as the cell. Antony van Leeuwenhoek, 1683, was first to discover or observe free cells like bacteria, protozoa, red blood cells, the RBCs, and the sperm. The cell theory: all cell organisms are composed of cell and their products. All plant cells have plasma membrane. All cells arise from pre-existing one. And the smallest living organisms are made of small cell, whether prokaryotes or eukaryotes. Unicellular and multicellular organisms. Single cell organisms are called unicellular. Organisms with multicellular organisms is called multicellular organism. Organism with more than one cell is called multicellular. Unicellular organism perform all kind of activities, and the surface area determine the amount of absorption. Some cells have micro. Cells are grouped together under three major categories. One is undifferentiated cell, one is differentiated, and another is dedifferentiated cell. Undifferentiated cells are the cells that undergo divisions and development. Differentiated cells, they are the post-mitotic cells that have undergone specialization or exhibit the division of labor. Dedifferentiated, sometimes what happens is the differentiated cells are capable of reverting back to undifferentiated cell, and this process they just lose their specialization, and it is called dedifferentiation. Types of cell are eukaryotic, prokaryotic. Eukaryotic are multicellular cells having a nucleus bound by membrane. Prokaryotic, these are the small cells that multiply very rapidly like bacteria or blue-green algae. Eukaryotic cells are the organisms consisting of one or more eukaryotic cells. Such cells contain membrane-bound nuclei as well as organelles. Animals, plants, fungi, referred as protista, are all eukaryotes, varying from single-celled organisms to truly multicellular organisms. Basic part of eukaryotic cell. Eukaryotic cells have cell wall. The plant cells have definite cell wall which enable cell lac, and the cell have fixed shape due to the cell wall. It is sufficiently strong, thick, and rigid. Plasma membrane. The cells are guarded by plasma membrane. It controls the flow of molecule to and fro of the cell, and the membrane is composed of lipid bilayer studded with structural protein. It's two-dimensional fluid or liquid crystal in which hydrophobic integral components such as lipid. The protein found on the membrane act as transporters and serve to carry molecules. It maintains cell environment. Cytoskeleton. The ability of the cell to change the shape and carry movements depend upon the cytoskeleton. It is a network of fibers running through the matrix of the living cell that provides surface of the chemical reaction. These are of three types: microfilament, microtubule, and what exactly it is? Microfilaments, it helps in the cell motion for the cell to adapt new shapes, maintaining structural integrity. Then microtubules, the network of proteinaceous cylindrical tubules. It is made up of tubulin molecules. It helps in maintaining cell shape by providing support, anchoring the cell membrane. These are involved in the cell movement and it participates in intracellular transport. Intermediate filament, these are the filament of protein fibers in the cytoplasmic matrix. They bind cells together at cell-cell junctions and form baskets around the nucleus, interweaving it. They are tough and durable. Endoplasmic reticulum, it is a network in the cell, in plasma of cell. There are two types, RER and SER. RER means rough endoplasmic reticulum. SER is smooth endoplasmic reticulum. These are rough endoplasmic reticulum are involved in active protein secretion, synthesis, modification, transport of cellular material. SER is smooth endoplasmic reticulum. They produce large quantities of lipids and have endoplasmic reticulum that is without the ribosome. What is the function of endoplasmic reticulum? The RER synthesis, serum protein, globulin, albumin, fibrinogen, membrane proteins of lysosome and the cell membrane. The SER synthesize lipids. The ER detoxifies the impure food and drugs that we eat. Released calcium ion helps in the movement of Golgi apparatus. It consists of stacks of flat membrane sacs. The Golgi apparatus serves to package the material for export and prepare for secretions. The packaging material moves from endoplasmic reticulum to Golgi apparatus. The proteins synthesized in endoplasmic reticulum are modified by adding specific group. Lysosomes. They are formed from leftovers of vesicles budded of Golgi apparatus. They are involved in intracellular digestion and are scavengers of the cell enzyme release digest cell particle. If these cells are released in the tissue, they may kill other cells and the cell in which they are stored, and they are known as autophagic vacuoles. Cell takes in material or give out or excrete. Endocytosis is divided into two parts like cell drinking, cell eating. Cell drinking is known as pinocytosis and cell eating is phagocytosis. So cell membrane invaginates or encloses the fluid droplet, or the solid particle membrane engulfs the particle and part of membrane pinches to encircle the particle within the cell. The ingested vacuole is termed as pinosomes or phagosomes. It is called phagocytosis. Exocytosis involves expulsion of the material out of the cell. The unwanted materials are leftovers called residuals. Cytoplasmic vacuoles, these are the voids in the cytoplasm that store small molecules like water, iron, sucrose, amino acid, whatever we eat. They appear bulbs and perform a variety of functions like concentrating mineral salt in plant cells or excrete material. Some primitive prokaryotes float in water cause of air and they are called Aerocures Ribosome. Their small complex structure, roughly 20 to 30 nanometer in diameter, consisting of two unequally sized subunits referred as large or small subunits that fit closely together. They synthesize proteins that form the membrane of secretory organelles and lysosomes membrane. Mitochondria, these are called as the powerhouse of the cell that contains their DNA. It has double membrane and the outer and an inner. Inner membrane bind mitochondria and inner membrane has many enfoldings called cristae. The enzyme an electron carries for the formation of molecules ATP. The cristae encloses the matrix that contains for TCA that converts all food to carbon dioxide, water and energy. Energy generated as electron is transported during breathing or cellular respiration. Plastids. They are self-replicating cytoplasmic organelles of the plant and algae cell. Chloroplasts contain pigment or starch or oil or a protein. They are divided into three classes: chloroplastids, chromoplastids, and leucoplastids. Plastids contains chlorophyll. It makes food. Yellow fruits have pigments called carotenoids, or also called chromophobes. Nucleus. It is the core of cell. It contains DNA and RNA. It controls all activity of the cell. It is storehouse of hereditary information to membranes. Nuclear envelope. This is for exchange of nuclear fluid and cytoplasm. The envelope disappears during the cell division and reappears. The site of synthesis of ribosome is nucleolus. Hereditary material is DNA. This is coil-like springs called chromosomes. Center portion is centromere. What are centrioles? They are the two small cylindrical cell organelles found near the nucleus. They are self-replicating, short fibrous rod-shaped organelles. They form cilia, flagella, astral spindle during division. They are made up of microtubules arranged as cylinder. They have cartwheel-like organization. Cilia and flagella, they are the projection from the cell, made up of centrioles. Cilia and flagella have the basic structure, but flagella are longer. They beat 10 to 40 strokes per second. The beating movement is called whip-like movement. Prokaryotic cells, they are primitive cells. It lacks nuclear membrane. They are the small cells that multiply. For example, bacteria, blue-green. So now the important one is difference between prokaryotic and eukaryotic. In prokaryotic cell, DNA is not coiled into springs but is arranged in circular form. Chromosome and microtubules are absent. Membrane-bound organelles are absent, like mitochondria are not there in prokaryotic. And prokaryotic cells are of two types. One is gram-positive, another one is gram-negative. Gram-positive, the stain is trapped by peptidoglycan, which form the outer layer of the cell, and gram-negative is outer membrane, prevent the strain from reaching peptidoglycan layer in periplasm. So now you can see the prokaryotic cell structure and eukaryotic cell structure is already given here. It is very, very important. You have to learn, and whenever there is a difference and you have a less time, you can, if you draw diagrams also, that is more than sufficient. But please, you have to remember this, because this is a basic structure. So everyone should remember the structure. And all the best for your exam. Thank you so much. Bye bye and take care.